many people still. It's great that we actually asked you to get the motivation letters done. Only so many motivated people. I mean, I swear to you, we have rejected 50% at least around that. So today we have our second day, and it is going to start with a plenary lecture of one of our plenary speakers from China. By the way, he's a Clarivate highly cited researcher, which is in top 1% of the most cited researchers of the living. And also there are going to be 3 sections, which are not parallel actually. We decided to not do that thing, to not split, BIOCON in different streams. And the parallel sections, which are not parallel but just straightforward sections, back-to-back lectures on agriculture and environment, on bioinformatics and genomics. It is going to be so inspiring. But before that, we have a very special thing to say. Because BIOCON 2023 family has a special hero today. And this hero is actually so committed to what we do that he decided. Can you think of what he did? He decided to spend his very important day with us, very, very far away. Far away, you cannot believe how far away. And this special person is having a birthday today. And this special person is none other than Professor Mirza Hassanuzzaman. Please, sing. Happy birthday to... Please come. Happy birthday to you. Happy birthday. Louder, louder. Thank you for spending this birthday with us, Mirza. It's such a pleasure. Thank you very much. This is your birthday cake, Mirza. We love you. On behalf of the BIOCON family, everyone is wishing Mirza the greatest birthday. Thank you for choosing this option to spend the birthday with us. We are so honored. Thank you very much. Thank you very much. Victoria will help you out with the cake. So this is how BIOCON 2023 turns into a really family holiday, almost like Christmas. Next time I'll probably do it during Christmas. How do you think of BIOCON 2023, or 2024 maybe, which would be dedicated to Christmas? That's great, that's great. But I can talk for ages, but now it's my big honor to introduce to this stage a great person, the Professor Mukesh Kumar Awasthi, to give a plenary lecture, please.
Good morning, everyone. Yes, sound good. So it's a very interesting session, and we would like to first, once again, to Professor Mirza, very happy birthday. God bless him, a very happy and prosperous life, and his family. So thank you, Professor Mirza, to celebrating with us your birthday. Thanks a lot, and thank you, Professor Misha, for nice introduction, a wonderful arrangement. And of course, I would like to first thank you very much his team, ITMO, BIOCON, and of course Stanford company, who has really wonderful organized this conference and make us possible to gather in this place. Thanks a lot, Professor Misha. Thank you. Yes, so sorry. Yes, I'm here. So I'm sure my name is Mukesh Kumar Awasthi, and I'm working in College of Natural Resource of Environment, China. This university is very close to the Xi'an, is old capital of China, and I'm sure all of you guys know Terracotta Army is very famous monument in China. So in future, if you have a time, most welcome to our university and enjoy the Terracotta Army, and of course you explored our university as well. Today I'm here going to talk about the organic waste biorefinery models towards sustainable circular bioeconomy, because the modern days we are producing huge amount of the solid waste, right? Yeah. So this is my content where I'm going to talk about very briefly everything, not talking too much because of the time limitations from the beginning. So this is the present scenario, what we are looking here, the waste and waste and waste. So the earth is only one planet where the waste is produced, and we all human is only one person who is responsible to produce this kind of the material. In our universe, none of the other planet is still known that where the waste is producing. And we human, of course, we have well-developed set up the mind, technology, many aspects we think we are well-developed as compared to the other. But still, we are looking a different way to sort out this problem, systematically looking the long-term way to sustain ourselves in this earth. None of the other people can sustain, only the human can sustain here. And we developed many of the technologies here that could be helpful to continue, recycle, and produce a variety of product from this waste material. So it's very important. In this regard, I would like to show you some pictures. I'm sure you all know that these are the lignocellulosic material, food waste material. And some of you just now, we finished our breakfast, morning breakfast, lunch, dinner. Every time we have a different kind of the materials as a waste. But I would like to change your perception that the waste, it actually is not waste. Think about carefully, is important resource. And we all researchers, if we change our perception toward to the waste and considering it's a resource to find out something interesting, but our knowledge, technology availability, all these factors involved to make us very difficult to be further recycled. Okay, thank you. Sorry. But we have a lot of the challenges, and this is the one of the challenge here. And also, the main aspect why we are considering this is the resource. The challenge is always there. That's why we're thinking to recycle this kind of the stuff, because it contains very essential biomolecules, and these biomolecules is necessary to be recycled in our ecosystem to maintain our resources. These are the another challenges. If the material is too lignocellulosic, is too complex, we have possible technologies, but still we are facing different kind of challenge. How to find out some interesting pre-treatment which can further and further use without increasing the total overall cost. Because most of the industries is struggling and they are putting 50% of the cost to find out something interesting pre-treatment technology. In a continue, I would like to say there's another kind of challenge. If we have the fruit waste material or food waste material, we have the different component. Yes, they are all essential for us. But in the meantime, we have certain things is trouble to us to recycle this kind of stuff. And this is the stuff. Most often we find it's very small concentration, but it's highly impact the overall microbial process. In a continue, yeah, there's not only one of the food or fruit waste material has such kind of the inhibitory component which influence the overall microbial dynamics and its transformation. But at the same time, they have many of the fruit and food material, and they have containing different kind of the inhibitory component, which make very difficult to recycle this kind of stuff. So in this aspect, we will look how we overcome and tackle these challenges. Yes, I very quickly summarize this point. We have to find out a variety of lignocellulosic material, how to figure out to recycle them, especially by finding the pre-treatment method. So in the further process, we make more smooth. The surface area for bacteria is also one of the major challenge. So these factors we are considering and think about to find some interesting pre-treatment method to remove such challenges. In this aspect, I would like to quickly describe to you that we have possible technology. Yes, we can remove these challenges, lignin, hemicellulose, cellulose component, hydrolyze them, try to make uniform site, find out something interesting physical, either chemical or biological pre-treatment method. But we try to combine all these technology. Or we find something very interesting here, cannot use single term. We try to use some microbial enzyme to treat this kind of this component and can further reuse in many attempts. But now the next question comes, what can we do with this kind of the stuff after the pre-treatment? Can we have possible a good technology or not? I would like to very briefly, this is our research work. We have done almost 2 decades in different aspects. But here I cannot cover up all of the aspects because of the time limitations. So I will very briefly explain each aspect. And if you have any question, please later on you can tell me. Here I would like to talk about when we have anaerobic digestion is in modern days, and actually is not modern days, you can say is the old and gold technology. It's a very old technology where we can use a different kind of the organic waste material and go through this 4-step process and always think about biogas. This biogas, of course, in old era is the major source of energy in different countryside. If you want more detailed information about this stuff, please go on this article. I'm sure you'll get more detailed and complete information. I would like to continue in here. Yes, this is another article you have to go through and get the more detailed information. I'm not going to explain too much about. I'm sure everybody know about the anaerobic digestion and biogas production. What I need to talk, the main aspect is coming soon, the dry digestion. We all know that anaerobic digestion requires more than 90% of water. But what do you think about the dry digestion? is something really interesting in modern days, because we don't have sufficient potable water. So how we consume for the anaerobic digestion purpose more than 90% water to get the energy. And we cannot get a sufficient yield of the energy by this process to maintain our customer requirement. So in this behalf, we try to integrate this technology, and this is our reactor where we are trying to reduce more than 60% of the water consumption, and at the same time we have the less water waste after the treatment. So it's really very interesting approach. You have much less slurry. It's also one of the non-digestive material residual waste. It contains different kind of the component. Yes, this is really very interesting. What we build, and I told you this reactor is built by waste material. Understand? Everything is the waste. Either this reactor is dry digestion unit, and of course this textile bioreactor to collecting the gas. Although we are working on the laboratory level, it's not come out on the market, but we have already sold this technology to the company. Sorry, is here. So we are looking how we can use a different kind of the waste material, textile waste material, and can develop a different kind of the reactor. It's really very great demand to recycle our textile material, because modern days everybody is too much fascinating. Every day change the fashion, and we are looking different kind of the different stylish cloth. So why not we use this textile material to develop something else? We just take this initiative. Yes, we try to conduct some experiment here. Use two different kind of the treatment, where the acclimatize and acclimatize treatment. Try to inoculate it with two different total solid material, as I mentioned to here. In a continue, this is some interesting result we obtained. And we looked at acclimatized conditions can help to enhance the overall biogas production. As you look at here, more than 82% as compared to the unacclimatized inoculum. It's really very interesting. In this way, reduce the water and get the better yield of the gas. But still, this technology took a long time to come on to the pilot level. This is the model of reactor, what I initially showed you in a previous slide. How we developed by the using of synthetic textile and plastic waste material. So here we are in color, and this is our inner side of the reactor where our material. By the using of this impeller handle, we rotate the material, and from here we are collecting the gas. Same time, we are running the other aspect. Sorry. Yes, is here. So in this case, we run, this is the total 9 liter working volume, is only 5 liter, is almost 50%. We have the total solid here, 22%. So this is our experiment design in continue. I tell you that we have 3 different organic loading rate and try to get something interesting byproduct by this technology. I would like to skip because of continuous, there's many information still waiting for me. And we talk about if the material is not biodegradable, or in comparison mostly cases for lignocellulosic material, is a low biodegradability. So what we can do such kind of the material? Can we integrate some other technology? Yes, we try here to integrate 2 different technology like anaerobic digestion process and thermochemical process. We skip the something in here, but find it, these both technology is not very effective or either is very expensive. We cannot afford. A lot of the maintenance cost is required. So in this regard, what we do, we have tried to skip some step, and here we integrate whatever the gas we produce by thermochemical process, introduce in our anaerobic digestion unit and get the biogas. But there is another problem here. Once again, our reactor very quickly dry. We cannot sometimes maintain the yield of the overall gas production. And the major issues, the microbial dynamics. The methanogenesis microbes who produce, they are not sustaining in long term. They always influence. And then there's a high chance this technology cannot succeed with this way. Then we took another concept in here, try to use the membrane bioreactor to recover some important nutrients from the gas, what we produce by thermochemical process. Try to use something interesting in here in the membrane reactor and use some kind of the nutrient media, cultivate the microbes, enzymes. We try different attempt with different aspect and try to recover. But still we are facing a lot of the challenges with this problem. So there is a next question here come out. If we always concern about the biogas, anaerobic digestion, biogas, anaerobic digestion, biogas, how long we sustain? It's like a monopoly, right? We not have any other option to produce any other byproduct. But in this behalf, we find something is really very interesting. We don't care about the methane here. Methane is not our main product in this process, because this process is always controlled by this last step, and the last phase is beyond to our control, because if the first three steps even is a very proper way is going on, but still we cannot balance the last phase. So we skip, we don't need the biogas. This is not our main primary product. We think about, we need the hydrogen, it's a much better clean energy. We need the volatile fatty acids because of this industrial market demand. And then we started something interesting to do. If you want more detail about this process, please go on this very important article and get the very detailed information about the mechanism aspect. In a continue, so as I mentioned to you, I don't care about the last methanogenesis. We don't need the biogas. We just try to boost these first three steps and get the better quality of the energy, better quality of the product. And here is our experiment design. We use the membrane bioreactor, and continuously we perform this experiment without disturbing the whole microbial mechanism inside the reactor. This is our reactor. So you can look, this is the membrane reactor and this is the membrane. We made it by polyethyl sulfate, and always here the microbes and nutrients continuously reacting. And reacting, sometimes we have to flush out this reactor to clean up by the back flush, but we don't disturb the whole mechanism. We continuously collecting here this is our something very interesting result. We try to use 4 different kind of organic loading rate and find some interesting result here that of course we have the higher amount of the volatile fatty acid, which is our main primary product, with the 8 organic loading rate, but at the same time we have also hydrogen. It's also very important for us because is a clean energy and much better than the biogas. So in this case, we almost say that we don't need the biogas. We can think about something else. And this experiment is really very successful. And we delivered this technology in Sweden, one very important biotech company. And he is using this technology to produce a volatile fatty acids. These are some important results we already published in renewable energy. Of course, the fuel. So if you want to more detail, please go on these articles, read, and understand the whole mechanism process. Now I'm switching something else also, which is directly integrated with our previous technology, where we can use this kind of food and derivative, what we have after anaerobic digestion get or before that. So the fruit waste is really important, and we often produce after eating the fruit. And a lot of the peels, apple peels, orange peels, I'm sure about 20 minutes before you have. So these are the important resource actually. As a researcher, it's very important for us to know is important resource. And we can make many byproduct. Understand? These are the things you think about, many anti-diabetic, obesity, there's many kind of the immunomodulatory antioxidant product we are thinking about to produce. And you know, I'm working in China, so there is a Chinese green tea is also very popular. I'm sure all of you guys know Chinese green tea is very famous all over the world. And after drinking that green tea, there is a lot of the waste material. You know, the green waste leaf after the extraction. That is also very important resource to produce something very interesting, because they contain very important biochemical product. We can extract by the using of biotechnology approaches. Here, these are something I would like to say. If you want to more detailed information, please go on these articles, read how we can use the fruit waste material to produce something interesting byproduct. Yes, this is another article. The Trends in Food Science Technology is really very good and high quality journal. We try to use the Chinese tea because we have many varieties of Chinese tea, and we produce the polysaccharide can be used for further for many medical beneficial product. In a continue, we have also, there's a many kind of the pineapple waste. If some guys and colleagues here from Thailand, Malaysia, yes, my friend is Misha is here, you know very well the pineapple is very important fruit, but I'm sure you know there is a 40% of the pineapple after scraping out is a waste, and only 50% we all guys eat. So it means each fruit come 50% is the waste and 50% we eat. So think about it's a really huge amount every day is producing. Yes, so we use this waste material as a resource and try to take out something very important byproduct, like as I mentioned, organic acid. We can recover some polysaccharide component. We also using something interesting by the thermal product and can use further this for the biopolymer production. I would like to show you. If you want to these details, please go on to these two articles. We have published in International Journal of Food Microbiology. So how we involving the microbiological technology to extract out something very interesting byproduct from these kind of resources. I would like to show you here the pineapple, how we can produce a various product. The main important component is biopolymer. So the biopolymer, after the extraction, biochemical product, we still have some non-digestive residual material. can further use. And we are trying to replace the plastic. Although this technology is not completely in a market, many of you guys are already working on different way in this way, using the biomass. But we have this important resource and we're trying to use different things to place them instead of using the plastic. We also try to produce some animal product so the animal can eat as a feeding material. We also try to develop something feeding for the fishes. So to cultivate the fishes and develop this product, animal feed product, fish feed product, livestock like pig, chicken. We also develop such kind of edible food product. Yeah, if you want more detail, please go on this article and read about how we can replace the plastic waste, leather, by the using of this technology, by the using of this important resource. Yes, there is something very interesting that we have to come out something bioactive compound by the using of biotechnology approach. So here we try to use microbes, try to apply something recombinant DNA technology. As I yesterday mentioned, we are struggling a lot of the food crisis. We don't have sufficient water to give the food, to cultivate the crops. Our soils are continuously deteriorating, as the Professor Mirza yesterday mentioned. Our fertility is continuously reducing. So a lot of the environmental stress is involved to reduce the overall yield and productivity of crops. So in this regard, we need to find and apply recombinant DNA technology can replace something interesting approach, can increase the tolerance limit of the plant, can recycle the different kinds of waste material and produce some valuable product. We try to gene editing by the different approaches and try to get different byproduct as antimicrobial, anti-cancer, antioxidant, probiotics. We apply this technology to enhance the microbial efficiency to get the quick byproduct. Yes, please go on this article. If you want more detail about these things, you can get very detailed information. I'm going to continue talking about the bioengineering. You know, the bioengineering in modern day is very important tools also, where we can use system biology, synthetic biology combined together and enhance the overall yield to produce something very interesting, nutraceutical probiotic component. Same time we can apply this technology Yes, we are applying same technology to produce the biofuel also. Try to enhance the tolerance limit of microbes. Try to increase its overall extracellular enzyme secretion efficiency. These are the important factors we consider to apply in the microbes and is increased in efficiency in the terms of the different byproduct. Yes, finally is conclusion. As I initially tell you, nothing is a waste. Everything is the best. Everything is important resource. But just change your perception, whether it's a waste or whether it's a resource. And then try to utilize your overall energy, knowledge, technology to get something very interesting byproduct in the long term to apply for the sustainable future for society. It's really important as a researcher to think about society, to get out something very interesting approach to help the society and this human can survive in long term in this earth without any interruption. So yes, thank you very much. These are my lab group members, and if you have any question, feel free to contact by here is my cell number, my email. You go on to my website also. These are my old colleagues. This is of course our university. So we are very happy to work together, and I'm very happy to all of you, anyone open if you want to any collaboration, discussion, any kind of things, whatever I can do for society, for you, and make a good friend. So most welcome. Thank you very much.
Thank you very much, Mukesh, for this friendly message, and now it's time for your questions. We have in this, please, in the middle, number 3, row number 3, Anna Kuznetsova, please.
Thank you for your presentation. Actually, I have several questions. The first one may sound naive. I was wondering why there is a difference between work volume and like original volume of the bioreactor that you invented. It's like 3 liters, yeah?
Yes, yeah.
Why this difference occurs?
You mean why the different reactor?
No. Why there is a difference in volume, work volume and
Yes,
the
okay,
volume
all right.
of the reactor?
Okay, now I understand. The total volume is the whole reactor capacity, but we cannot work in the whole reactor capacity because we need to give space to be anything such as some gases produced, or you need to have some rotations overlapping, sometime. And if this thing, if you fill up the complete overall efficiency of the reactor, it's come out something. And it's high risk also to damage the reactor. So this is a very important consideration you should apply for your research. From the beginning, you must remember these things. All right?
Yeah, thank you. And the second question actually about fungi leather. You showed the article about fungi leather, and I'm interested in which fungi did you use?
We are using Ascomycetes, yes, Ascomycota. Yes, it's very important, especially Rhizospora, Rhizospora.
I suppose.
Yes, Rhizospora is very important fungi, and it's very good for this purpose. Understand? Yes. And also a lot of the mushrooms is also belong to the Ascomycota, so this also can think about.
And as a substrate, as a waste, you used pineapple. Yeah?
Yes, of course.
Yeah,
yes.
Thank you a lot.
We have a question by my right hand, please. Row number 2.
Thank you for your presentation, and I wanted to ask you if you try to derive these technologies to the industry in a big scale, and if it's more expensive for companies to reduce food waste as usual, I mean burning it or something else, or to recycle it to something else.
Yes, actually, we are working on this aspect to recycle the fruit and food waste material, because most of the cases this material goes for the thermochemical process. So in these cases, the thermochemical process is required more energy to be incinerate the stuff, and in this case you cannot properly recycle all the nutrients they contain. So we try to develop something to take out from this kind of material, as I mentioned to you, some antioxidant component, and thereafter some indigestive material left we try to use for the, as I mentioned, for the fungal leather production, and also some kind of the other biopolymer production, such cases. And there is one more interesting thing we also apply, some post-consumption waste and pre-consumption waste. So we use the pre-consumption waste to develop something like a vegan food. Modern days, a lot of the people is a vegan, so we are trying to develop something vegan food, but initially now we only for the animal feeding purpose, not for human purpose, because we have a lot of the approval, and then we can further use for the human purpose, but still we are trying to apply for the animal feeding purpose, this thing. Yes.
Okay, thank you. And so the second question, you tell about the plant waste.
Yes.
But can you tell something about animal waste? I mean, food, animal food.
Animal food waste or animal livestock waste?
Yes, yes.
Okay, all right. Yes, so livestock is also a very favorite subject for me as a researcher. We are working on manures, different kinds of manure, like cow manure, pig, sheep, all kinds of the stuff manure. And my university has a lot of the farmhouses, and we are producing huge amount of this kind of organic material. So we use mostly cases the anaerobic digestion process first, and then whatever the residual waste left, we use for the composting purpose. And then we can give to the farmers this compost as an organic fertilizer, because all over the world nowadays thinking about organic farming, and even my own university in China government is also forcing to go ahead for the organic farming. So we use this compost for the apple farmhouses to enhance the soil fertility, soil nutrient level, such kind of things. Yeah.
Okay, thank you.
Yeah.
Probably no more questions. Well, it's great to know that. The final question probably, right? By Paulina. You should tell now if you want any more. Question number 4, please put up your hand right now. If there's going to be any question number 4, 3, 2. The last question number 4, okay. The next question on the last, please, Paulina Shebekova.
Mukesh, thank you for a wonderful presentation. And my question to you is, how people can recycle the waste at home conditions? I think that if every person can recycle the wastes, the amount of these wastes will much less. Maybe can you give some tips for it?
All right, thank you, Paulina. Oh, very nice question. Yes, it's really very important. On a source level recycling, where the waste is generating. So the first thing, the awareness is very important. Paulina, I told you, understand? So the people have their own responsibility toward their own waste. They don't think that the waste generation is their responsibility, but because we pay to recycle, so we don't have any other further responsibility. So this is the first thing. The awareness is very important, what you raise the question. And the next question here, that how you can recycle. So there is one very important, the Australian company is Bolasica. His name is Bolasica, understand? He has a small composting unit. It's a very small container, like your dustbin is here. A small dustbin where you can keep your fruit waste or food waste material in a different layer. And they also provide very important microbial consortia. So you can put a fruit layer, there is some certain microbial consortia layer, once again, and it's within the 20 days, as per their report, you get the compost. So in these cases, your own food and fruit waste can recycle in your own home, right? Yes. And the second point, as I mentioned, the awareness. In Sweden, I told you, I very admire this country because they have very interesting system for the recycling. Such as we are using the plastic bottle to drink a water, understand, and we throw it, most often in the dustbin. But suppose you think about what you have the plastic bottle, you buy $2, $1, suppose $1. And I commit you, after finish your water bottle, give me this bottle, I will give you $1 return back. Right? So I'm sure everybody, after finish your water or beer or whatever you drink, soft or hard drink, you will never throw your glass and plastic bottle. Everybody should be kept properly because it's your money, right? And in these cases, we go. In Sweden, there is a certain market where you go buy your stuff. So in front of that shop is one machine. Put your stuff, how many bottles? Take out your money instantly. So you happy? Yes, waste is also recycling. Everybody happy, right? Correct? So this is the one system, and we should aware about how to we think about carefully for our nature. We should identify our responsibility toward to the nature, and waste recycling. Yes.
And we also
Thank
have
you.
question number 4. Question number 4, which is going to be the final question for this section, please. Third row from the left.
Thank you so much for the presentation. I actually have a question about biogas.
Yeah.
So it's all that it's not the main product that we use, and we stop on the hydrogen and the VFA.
Yes.
So how do you inhibit and control the synthesis of biogas?
Okay, you mean, oh yes, I understand you mean how we control to the switch the step of the third to the fourth step. We try to use some inhibitory component actually, I told you. We have already, I show you here one paper, so you read the information, you may get the complete information. We use some important inhibitory component can interrupt you to switch on to the methanogenesis step. Listen. So that is our input. Yeah.
Thanks so much.
Welcome.
Thank you very much, Mukesh. It's great that you came to BIOCON 2023 to become a plenary lecturer. And once again, let us overwhelm Professor Mukesh Kumar Awasthi with ovations. Thank you very much. Thank you very much.
Thank you very much. Thank you.
So before we go to have a break, and I swear to you, at 11:15 sharp, we are going to start the next section. So I mean, just believe me, we are going to do that, because out of respect on behalf of organizers towards your time. But before you go, and you're going to have a 15-minute break, we have prepared a special surprise for you, please. And in order to experience that surprise, please, your eyes on the screen.